Good morning, everyone. Welcome to the webinar on analysis of Ion Torrent data in Avadis NGS. The Avadis NGS product has been out in the market for more than a year now. The goal of the product was to allow end users to carry out analysis in an interactive and user-friendly manner. The Avadis NGS product is built on the same underlying platform as GeneSpring. So those of you who are GeneSpring customers will find the look and feel very similar. Right from the first version of the product, Avadis NGS supported import of aligned files which are in industry standard SAM, BAM or Illumina specific ELAN formats. The workflows that were there in the earlier versions of the product were ChIP-seq, RNA-seq and DNA variant analysis. With version 1.3, which was released last month, we also added support for small RNA analysis. In addition, we now provide alignment within the tool. You can align small RNA reads or DNA reads within the Avadis NGS aligner, which is called Cobweb. So the typical NGS data analysis pipeline is as follows. We have a sequencer which generates reads. The sequencer comes along with some software. Sometimes the software goes as far as aligning the reads against a reference genome and generating SAM-BAM files. Sometimes the software goes further. It can carry out tertiary analysis. It can analyze the aligned read files and call out variants, or it might carry out quantification, etc. So the Avadis NGS product supports import of multiple of these files. If SNPs have been called and are reported in VCF format, you can import the VCF files into Avadis NGS. Similarly, if there are quantification results in a TSV format, you can import those results into Avadis NGS. Moreover, you can import the aligned read files which are in SAM-BAM format and carry out either the SNP detection or quantification steps within Avadis NGS itself. And finally, since we have now introduced alignment capability to the product, you can start off with FASTQ files and perform the alignment within the tool before carrying out the SNP analysis or quantification, etc. So let's see what Ion Torrent data looks like. On the Ion Torrent community website, a lot of the data sets are made public. So we have data sets which were generated from the Ion 314 chip, data sets generated from the 316 chip, and those generated from the 318 chip. Clicking on any one of those links takes you to a full-fledged page which describes that particular data. For instance, here is the page which describes 2318 human resequencing runs. Now most of the files are in standard format. The aligned files are in BAM format and the files have been aligned against the HG19 reference. The SNPs are present in VCF format, etc. So all of these files can be imported directly into Avadis NGS into a DNA variant analysis experiment, and you can verify the VCF contents, or you can carry out SNP detection and compare the two different SNP results, etc. So all of this has been covered in other webinars which have discussed the DNA analysis pipeline, webinars where the small RNA analysis pipeline were discussed, etc. So today I will discuss only those data sets which cannot be imported directly, but a little bit of processing is needed before they can be handled within Avadis NGS. So as I was mentioning, if you have contents of this form, you can either start with the FASTQ files, align them within Avadis NGS and then carry out the analysis, or you can start with the BAM files. You can import the BAM files and carry out SNP calling on the BAM files, or you could start with the VCF files and verify their contents. But some of the other data sets are not handled in as seamless a manner. For instance, the recently introduced AmpliSeq Cancer Panel datasets. If you were to click on that particular page, it shows you the full details of the dataset. And the item to note on that page is that the reads are not mapped against the HG19 reference. They are instead mapped against the AmpliSeq Cancer Panel reference. In other words, the reads are mapped only against the sequence of the amplicons. The sequence of the amplicons is given as a FASTA file, and the locations of the amplicons are given in the form of a bed file towards the bottom of the page. So if you want to carry out analysis of these data sets within Avadis NGS, what we need to do is we need to convert these files into chromosomal coordinates. So some of the issues that we'll discuss today are how to load these amplicon aligned reads into Avadis NGS, how do we verify the amplicon coverages to see if they have sufficient depth or not, and since the amplicons are given as a simple bed file, a 3-column bed file with chromosome start and end, how do we know what does each amplicon stand for? How do we find out which gene and which exon does each amplicon correspond to? Once we import the Ion Torrent predicted SNPs, how do we verify them within Avadis NGS, how do we carry out downstream analysis on it, and finally the AmpliSeq Cancer Panel itself. It consists of a list of around 790 variants which have been identified by a panel of cancer experts. So given this cancer panel, how can we in 2 clicks verify whether a DNA sample has any of the mutations which are listed in this panel or not? So that is what we'll do towards the end. So let's start by importing Amplicon aligned reads. The reads have been aligned against the Amplicon sequences. So what we need to do is we need to take the Amplicon specification, which is present in the bed file, and we have a Python script which will then convert the SAM file into genomic coordinates. So this script will be made available on our website and on the Iontorrent community portal. Once we remap the reads into genomic coordinates, it will allow us to visualize the reads in the context of other annotations. We will be able to see the reads in the genome browser with all of the other tracks which are there in genomic coordinates. And that Python script is a simple script, and after we run it from the command line, we now have a SAM file where all of the coordinates are genomic coordinates. So using this file, we can create a DNA variant analysis experiment which use the reference, which is the human HG19, and all of the analysis carried out within Avadis NGS is within the context of genes. So we make multiple gene annotations available through our annotation server. The user has a choice of performing analysis against RefSeq genes, UCSC genes, or ensemble genes. In this particular case, I've chosen RefSeq genes and specified the library layout, which is single N. After that, I choose the SAM file which I created using the Python script from the Amplicon aligned file made available by Iontorrent. We have an option of changing the sample name at this point. After we do that, the data gets loaded into a DNA variant analysis experiment, and this is the first view we have of the experiment. The layout of the tool is a standard 3-panel layout. On the left, you have a project with all of the experiments in the project. Below that, you have the open project, open experiments. In this case, I have only one experiment open containing the sample I just imported. The center panel shows the genome browser. It has one track corresponding to the sample that I just loaded. We can immediately see that there are at least 2 different amplicons in this particular chromosome. The right hand side is the workflow with quality inspection steps at the top, filters and analysis steps following that. Typically one works their way from the top of the workflow towards the bottom to get the desired results. So now that we have those 2 peaks, we can verify them in the genome browser, or we could first check what the different quality attributes are of the reads that we just imported. There's a whole lot of QC metrics which are displayed in a nice visual form. I won't go into all of them. We can look at the base quality and the read quality. A lot of the bases seems to be of excellent quality, around 29. The read quality is simply the average of the base qualities of all the bases within a read, and that's also quite high, around 28. We can then get on to verifying the amplicon coverages. There were around 190 amplicons in the bed file specified on the Iontorrent website. We can try to see do they have sufficient coverages or not. One way of verifying the coverages is to simply zoom in to the location on the genome browser. So this is, for instance, the very first peak that we noticed on the genome browser. It is on chromosome 1 around the 43 megabase pair location. And if you zoom into that particular section, we can see that it's actually the 10th exon of the MPL gene. So one could go through the genome browser looking at the high coverage peaks and try to infer what the amplicons are. But what we would do more naturally is to simply import the amplicon specification from the bed file provided by Iontorrent. For that, we go to the utility section of the workflow and click on the import region list workflow step. We choose the bed file. We had to make some slight changes to the bed file because it was incorrect. We had to add one extra region which was not present in the bed file. So after we import that bed file, it shows up in the navigator as a region list, and the region list is indicated by this icon out here. Given a region list like this, we want to go through the genome browser looking at each one of the regions that are present in this particular region list, because each region corresponds to one amplicon. For that, we simply have to right click on the region list and say navigate in genome browser. So you can see that the navigate spreadsheet in the genome browser is active, and all of the 190 amplicons are shown as a spreadsheet out here. By simply double-clicking on any row in this spreadsheet, Genome browser will zoom to that particular region. You can see that you have a gray area marked in the Genome browser out here. That area demarcates the boundary of the amplicon which is highlighted in the spreadsheet. There are multiple tracks in the Genome browser which help you interpret what is going on in that amplicon location. You can see the read track out here. You can see the coverage is very high, 1470 approximately. You can see the dbSNP information. There are 5 dbSNPs in this particular location. You can see multiple transcript tags. Even though we started our analysis with RefSeq transcripts, you can use the annotations which are there out here in the Genome browser to load other transcript tracks purely for visualization purposes. One can also see a mismatch histogram out here that indicates locations where a substantial number of reads are differing from the reference, and the mismatch histogram gives an indication of what are the possible variant bases out there. The Genome browser supports a very good zoom functionality, so one could try to zoom into that particular location to see the bases in more detail. As we do that, you'll notice that these are all Cs out here, but some of them are brighter than the others. That's because density of the base indicates the quality of the base. By hovering the mouse pointer on any of those bases, we get a tooltip which provides comprehensive information at the base level and at the read level. We can see that some of the bases have low quality. For instance, this base has a quality of 5. So before carrying out the coverage calculations, we would like to remove the poor quality reads and then only calculate what is the high quality read coverage. For that, we can carry out filtering. We provide a comprehensive filtering step with all kinds of metrics that you can choose. Here we're using very strict filtering criteria. We are filtering out reads with average base quality below 20 and mapping quality below 70. I didn't show you the mapping quality even though it is there as a different QC picture, but the quality was going between 0 and 100, and 70 was actually pretty decent, but we were removing reads below that. Once we carry out that filter step, it shows you how many input records were processed and how many of them were filtered out because of this criteria. You can see that out of 535,000 reads that were processed, 48,000 have been filtered out. So that's approximately 8 to 9% of the reads have been filtered out. And this is the read list inspector. It shows you that in the read list how many reads are falling against each chromosome and how many of them are aligned in the positive direction, negative direction, etc. So this is the read list that we'll be using for subsequent analysis. So now let's get on to target region QC. We saw how we can verify the coverages visually. We can just jump from one amplicon location to the other, noting the coverage in the Genome browser. But a more quantitative way of verifying that is to perform targeted region QC, which is present in the quality inspection section of the workflow. When we click on targeted region QC, we choose what are the set of regions for which we would like to find out the coverages and what read list should be consulted in computing those coverages. The padding option, which is out here, allows you to also analyze coverages in a slightly expanded section of the region. In our case, we will be setting the parameter to 0 because all of the reads were aligned only against the amplicon regions, so we don't expect to have any coverage at all outside the amplicon regions. Once we finish the targeted region analysis, we get another region list in which each of the regions that we're analyzing has been annotated with targeted region QC metrics. So 3 extra columns have been added to each region. The first column indicates the GC percentage in that region. The second column shows the number of reads that are aligning against that region. And the third column indicates the average depth of coverage in that region. You can see that the average coverage is quite high. But going through the spreadsheet manually inspecting these 190 rows and trying to infer patterns is a little bit cumbersome. So what we can do is open this region list in the region list operations viewer, and there we can try to see if there are any patterns between the different columns that were added by the targeted region analyzer. So for instance, one common question is there a GC bias? So we can draw a scatter plot between the GC percentage and the average coverage columns, and we can see vaguely that there is some GC trend. At the very lower extremes of the GC percentage, the average coverage is on the lower side. At the very high ends of the GC percentage, the average coverage is a little bit low. But when you're seeing all the points out here, it's hard to figure out exactly what is the coverage in these locations. So you can always zoom into these regions, and the region list operations viewer is a very full-fledged viewer which will allow you to do all these operations. So if we zoom in out here, we can see that there are actually only a handful of regions which have average coverage below 300, and only one of them has coverage less than 100. We can click on it and see what is the ID of that particular region. It happens to have ID 236,158, and that was an ID in the Amplicon file given by Ion Torrent. If you want to create a list of all these regions which had less than 200 coverage, again the region list operations provides a very nice filtering mechanism on the right hand side. We just choose all the different filters that we wish to set. In this case, we want to find out only those amplicons which had coverage below 200. So we set the appropriate filters, and we can see that there are only 5 such amplicon regions. We could save these regions into the genome browser and explore those into the navigator and explore them in the genome browser like we did with the earlier region list. So far we have seen how to import Amplicon data into the tool and how to verify the coverages. But what we need to do is find out what gene does each amplicon correspond to and what exon of each gene does it correspond to. So we need to annotate the amplicons. For that we'll be using a script that is included with the tool. The tool has a script editor through which users can write scripts, but we also make available scripts that we think will be of use to many users. So we just click on the script editor button in the tool and shows the list of all the scripts that are packaged with the tool. If we click on the find exonic regions, it will go through the transcript model that was selected at the time of experiment creation. In our case, that was RefSeq genes. So it goes through all the RefSeq genes and creates a list of all of the exons in the navigator. So now what we need to do is take our targeted QC report, and for each one of the amplicon regions, we need to find out what are the gene ID and gene name of the overlapping exonic region. Once we do that, the targeted QC report, which earlier had 7 columns, if you remember, it had 4 columns to start off with. That was the amplicons that we imported. We added 3 additional columns to it by doing targeted QC report, and now we had added additional columns through it via the annotation mechanism. So each one of the regions has been annotated by what fraction of that region overlaps with an exon, how many base pairs of that region overlap with an exon, what are the start and end coordinates of the exon which overlaps with it, and which gene does that exon come from. So this pretty much constitutes all the annotation that we need for these amplicons. We could try to do some summary statistics on it. For instance, it is interesting to find out if multiple exons of a gene are being covered, or if only a single exon has been chosen from a gene. So we can again go back to region list operations and try to summarize the gene name column, in which case we get a distribution like this. We can see that there are a lot of exons of the ATM gene, but there are only a couple of exons of the MLH1 gene, etc. So far we saw how to verify the coverages of the amplicons. We saw how to annotate the amplicons. So now let's look into the SNP results that were predicted by the Ion Torrent server and how to import those SNPs and make sense of them. The SNPs were present in a VCF format, so we simply need to choose the VCF file, and when we import a VCF file, we add additional information to it from a read list. So we also choose a read list with which to annotate the VCF file, and as I mentioned earlier, we are choosing the read list which we got as a result of the filtering process. Once the VCF file is imported, it shows up as an object in the navigator, and you'll notice that the object has a special icon. It looks similar to the region list, but there are dots instead of a line out there, indicating it's actually a variant region list. If you double click on that variant region list, it shows an inspector. It shows information from the VCF file, the header part of it, and there are actually only 2 SNPs which have been called by the Ion server. So both of those rows are shown out here. Most of the information is directly from the VCF file, but 4 columns are added additionally during the import process. What we do is for each one of the variants, we consult the read list which was selected and find out what is the fraction of reads from that read list which are supporting that variant call, what is the total fraction of reads which are differing from the reference, and the total number of reads which are overlapping the reference in that particular location, and we also compute the strand bias. It's a number between 0 and 200, and something close to 0 means there's almost no strand bias. So as you can see out here, both of the SNPs which have been called are heterozygous SNPs because they're 50% supporting reads for the variant allele, and the strand bias is very, very low. So both of these would be considered high quality SNPs. We can also inspect the SNPs in the genome browser. Just like before, we can drag that region list onto the genome browser and look and double click on the substitution that was there. We zoom into that particular location and we can see it's almost a perfect heterozygous location, and there is a DB SNP ID corresponding to it. The coverage here is very high, it's in the 3000s. So if you want to compress all of the reads to get a better idea of what the read patterns are in this particular vicinity, we can launch the variant support view directly from the genome browser. The variant support view takes a vertical slice of the genome, typically 10 bases on either side of the point that you selected. So it takes the 21 base pair window and takes that vertical slice through all the reads, and all the reads which have exactly the same sequence in this 21 base pair window are grouped together into a single cluster. So you can see that there are 5 clusters out here which have size at least 5. The top 2 clusters dominate. The first cluster corresponds to the variant allele because there's a G in the reference and there's an A for all the reads in that particular cluster. The second cluster matches the reference allele, it has a G. Then you have some few smaller clusters of size 16 and 10 and 6 and so on. If you want to eliminate the smaller clusters, we simply need to change the minimum cluster size, and then we get only 2 dominant clusters. So this is a very easy way of verifying the cleanliness of the neighborhood of a SNP. So now we know how to import SNPs and how to verify them visually in the genome browser, both by looking at the read track and by consulting the variant support view. Once we have high confidence SNPs, we can then focus on what is the downstream effect of these SNPs. What effect do they have on transcripts and protein sequences? For that we have a SNP effect analysis workflow step. When we click on that, it asks for all the different effects that we would like to predict. By default, all of them are tick marked. Once we finish the SNP effect prediction, it shows 4 rows. That is because the second SNP that we had in the imported file occurs in a location where there are 3 overlapping transcripts, and it shows an effect on each one of those transcripts. The effect happens to be exactly the same. It is a synonymous SNP and the amino acid does not change, but the position of that variant in the transcript is different because the cDNA sequences are different. Okay. So this is the SNP effect prediction workflow step. In this case, we had only 2 SNPs and both of them have exactly the same effect. But you can easily imagine how if you have hundreds or thousands of SNPs from a whole genome analysis, you could run SNP effect prediction and then filter out and retain only the non-synonymous coding SNPs or frame-shaped SNPs and focus on them for prioritization purposes. So far we have seen how to import the SNPs predicted by the Ion server and how to verify them. You could also try to carry out SNP analysis within the tool itself using the SNP algorithm present within Avadis NGS. For that, you would click on the SNP detection wizard, you would choose the different cutoff criteria, and you could try to verify the SNPs that have been called by Avadis NGS. You'll notice that there are some very clean deletions which look quite plausible. Obviously, you'll have to verify the strand bias and all the other supporting information, but this deletion looks like a possible deletion event. Similarly, there are other homozygous substitution events which have not been called by the Ion server, but based on the mismatch histogram and the cleanliness of the read track have a possibility of being very correct predictions. So we've seen how to import a VCF file and to analyze it, and also how to call SNPs within Avadis NGS itself. So now we go back to the comment I made right in the beginning of the presentation. The whole goal of the AmpliSeq Cancer Panel is to check whether samples have known mutations. The panel is a collection of 790 odd mutations which are considered to be very important for cancer, and they cover 46 oncogenes and tumor suppressor genes. The panel itself as a mutation list is available as an Excel sheet. The question we wish to answer is which of these mutations are present in a particular sample. This can be done in exactly 2 steps. We choose the Ion panel. We have done some transformation to convert it from an Excel sheet to a TSV file. This particular TSV file will make it available on our servers. Once we choose that panel, we choose the read list against which we want to verify it. And the very next step, each one of the variants mentioned in the panel is annotated with data from the sample. The important columns that are added are: what are the percentage of supporting reads for the allele, what are the variant reads which support something other than the reference base, what are the total number of reads covering this particular location, and what is the strand bias at this location. So once we have this list, if we sort on the supporting reads percentage column, we can see that there are actually only 3 rows which have any meaningful amount of supporting reads. The very first row, that particular allele has 49% supporting reads, and the strand bias is very low. This is in fact one of the SNPs that was called by the Ion software. The second row, there are 38% of the reads are supporting the variant allele, but unfortunately the strand bias is quite high, so it is there's a possibility that this is a false positive. And the third row is really along the borderline. Only 13% of the reads out there are supporting this particular variant call, and the strand bias is very high. So you would not really consider this as a correct call. So in other words, we have seen in just 2 clicks how to verify that there was exactly one SNP of all the 769 variants mentioned in this panel in this particular sample. So this is the way in which a typical sample could be processed. You would have the Ion panel, and you would simply import the Ion panel and the patient sample. You would take a look at this list, you would sort on supporting reads and see how many of the rows have high supporting read percentages and low strand biases. We spent a lot of time on amplicon analysis. We'll take a brief look at some of the other analysis present in the tool. Small RNA dataset has recently been made available by Ion in Feb of this year, in fact. If you click on that, you get details about the dataset, and you'll notice that the reads here have again not been aligned against the reference genome, but they've been aligned against the precursor miRNA genes and the tRNA genes. The reference file is not the HG19 reference, but rather it is the sequences comprising all the small RNA genes and the tRNA genes. Again, as we mentioned earlier, these files are hard to view in a genome browser, and we'd also like to view them in the context of other annotations. So we use the same mapping script that I mentioned earlier on to convert this file into genomic coordinates. Only difference is that in the case of the Amplicon Data, the Amplicon bed file which specified the location of the amplicons along the genome was provided by Ion, whereas here that bed file is not there. So we simply take the annotations which are part of the Strand small RNA annotations and create a bed file out of that and use that for mapping purposes. Once we do that, we can convert all the 5 samples that are part of this data set. We can load them into a small RNA experiment. We can create experimental groupings as specified on the Ion web page. 3 of the samples are lung samples and 2 of them are placenta. We can create interpretations and find out the differentially expressed genes. You see that a lot of genes are both upregulated and downregulated in placenta with respect to lung. We can verify these results. We can view these results in the small RNA gene view. It's a powerful gene view which shows the small RNA structure out here. It shows the locations of the mature miRNAs. You'll notice that some of the bonds are connected by blue and some of the bonds are connected by red. Those are the locations of the 2 mature miRNA locations. Those locations are also shown in a linear manner out here. The entire precursor gene sequence is shown. The structure of the gene is shown in dot parenthesis notation. The locations of the mature miRNAs are shown in red and blue out here. And you can see for each sample how many reads are aligning against the different portions of the precursor gene. Out here it shows that all of the reads aligning against the precursor gene are aligning only to this mature, are originating from this mature miRNA, whereas the other mature miRNA is not contributing anything at all. So those things are possible through the small RNA gene view. And in addition, you can see mismatches against the reference genome, and it's possible that those are because of RNA editing. So small RNA analysis is a very comprehensive analysis. It allows for discovery of novel genes. It allows you to validate the annotations and perform all forms of differential expression analysis as well as target prediction analysis. But all of these details are covered in the small RNA webinar, so I will not be going into further detail. I'd like to briefly mention the new alignment features. There's full support for Ion Torrent reads. We support variable length reads of any length. We have had great success in aligning reads 400 length, 500 length, etc. We can support arbitrary number of gaps, and there is robust handling of multiple matches. There are comprehensive trimming options. The trimming options change depending on the type of data that you're aligning. If you're aligning small RNA data, then 3' adapters might still be present along with the read. So there's an option to remove the 3' adapters by giving the adapter sequences. If not, you can trim either a fixed number of bases, or you can trim based on the quality before alignment. In fact, you can also trim after alignment depending on the alignment score. With these options in place, we have verified that the alignment that is carried out on the Amplicon data, it matches extremely well with the aligned file that was provided on the Ion community website. We also allow for screening databases. We make available some screening databases. If the user wishes to screen against their own proprietary sequences, they simply need to upload a FASTA file and call it a screening database. It will show up in this particular dialog box. And once the alignment has been carried out, users can create an analysis experiment from the alignment experiment and carry out all the DNA variant and small RNA analysis that I showed you some time ago. So that brings us to the end of the webinar. If you want to try out Avadis NGS, you can simply visit our website www.avadis-ngs.com. You can register on the website and download installers.